Welcome to my poster. We know proteolytic cleavage is a crucial process for many positive sense RNA viruses, including the coronaviruses. They have both their own viral proteases, as well as cleavage of viral proteins by cellular proteases. And we know that these processes are absolutely essential for efficient virus replication. For this reason, we sought to characterize the different cleavage events occurring within SARS CoV-2 infected cells. For this, we used a mass spectrometry-based approach termed N-terminomics. This enriches for protein N-termini, both native N-termini and those generated through proteolysis. We performed this in A549ACE2 and VIRO-E6 cells across an infection time course, enriching for protein N-termini. From this data, we identified at late times post-infection a significant number of peptides corresponding to proteolytic cleavage events. These included viral proteins, highlighted in blue, as well as a subset of cellular proteins. We identified a number of novel viral cleavage sites, including many sites in the nucleocapsid protein, in ORF3a and spike, amongst others. Of particular interest in spike were the signal peptide cleavage site, which is pyroglutamate modified at Q14. And we also identified a new cleavage site at 637 in both Viro and A549 cells on a flexible loop near the furin cleavage site. And we found that this is sensitive to cathepsin inhibitors. We also identified cleavage of a range of novel cellular proteins by the papain-like and main proteases. We saw temporal regulation of these cleavage events, and when we proceeded to perform siRNA knockdowns, as you can see in panel A, a majority, though not all, of these proteins reduced viral RNA copies following siRNA depletion. But all of these substrates showed a significant drop in viral titers following siRNA depletion, suggesting that these are actually essential for efficient virus replication, and that proteolytic cleavage is not simply to turn off the function of these proteins. And we also found, using currently available inhibitors, that a subset of these proteins, so the kinases SARC and myosin light chain kinase, had inhibitors, and that the use of these inhibitors reduced levels of infectious SARS-CoV-2 at low to sub-micromolar concentrations. I'd like to thank my co-authors. If you'd like to hear more on this story, please read our preprint, and thank you to the funders.